Myself Siddiq Kaur, Associate Professor, Department of Pharmacology, Jyoti Devilal College of Pharmacy, Haryana. Dear students, today we will discuss about cell injury, adaptation and cell-death. So here are some key concepts. First one is homeostasis. It is a self-regulating process by which biological systems tend to maintain stability while adjusting to conditions that are optimal for survival. Next one is adaptation. It is the ability of body to cope up with the stress. So we can say change in cell morphology and function in response to a stimulus. It is a reversible process. In three types, ability of body to cope up with stress. First one is increase in stress, second one is decrease in stress, third one is change in stress. We will discuss about adaptation in our other slides. Third one is cell injury. Too much stress exceeds the cell's adaptive capacity. It is of two types, reversible injury and irreversible injury. Reversible injury is non-lethal and irreversible injury means cell death. Cell death is of two types, apoptosis and necrosis. We will discuss about it in detail in our next slides. Homeostasis. Homeostasis refers from the Greek words homeo and stasis. Homeo means similar and stasis means stable. So it is a self-regulating process which has the ability to maintain a constant internal environment in response to the external environmental changes. Control of body temperature, blood pressure, acid-base balance are few examples of homeostasis. It is of two types, positive feedback homeostasis and negative feedback homeostasis. Now let's have a look on positive and negative feedback homeostasis. Positive feedback homeostasis supports the stimulus and increasing the productivity. This type of feedback mechanism promotes the change to proceed further. In negative feedback homeostasis, the effect of stimulus is reduced and this will decrease the productivity. And this type of feedback mechanism stabilizes the biological system maintained under homeostatic conditions, and it is important for a constant internal environment. So, blood clotting, childbirth in mammals are example of positive feedback homeostasis, and regulating temperature, blood pH, hormone levels is an example of negative feedback homeostasis. Students, here is the chart which represents the differentiation between both feedback mechanisms. In positive feedback mechanism, it results in the amplification or growth of the output signal. While in negative feedback mechanism, it results in the inhibition or slowing down of a process. Positive feedback homeostasis supports the stimulus and increasing the productivity, while negative feedback homeostasis reduces the effect of the stimulus and decreasing the productivity. Positive feedback is less frequent process and negative feedback is more frequent process. Positive feedback is less associated with the stability of the system, while negative feedback is more associated with the stability of the system. Examples are blood clotting and childbirth in positive feedback homeostasis, and in negative feedback homeostasis, the control of optimal internal environment by regulating temperature, body pressure, blood pressure, pH, hormone level, etc. Cell adaptation to injury. There are 5 cellular adaptations to injury. First one is atrophy. Atrophy means there is decrease in size of the cell. Hypertrophy means there is increase in size of the cells. Hyperplasia, when there is increase in number of cells. And metaplasia means when there is change in particular epithelial tissue. Dysplasia is the abnormal development of the cells. Adaptation. Adaptation is of 2 types: physiological adaptation and pathological adaptation. Physiological adaptation represents responses of cells to normal stimulation by the hormones. For example, the hormone-induced enlargement of breast and uterus during the pregnancy. Pathological adaptations are responses to stress that allow cells to modulate their structure and function and thus escape the injury. So, there are 5 types of cellular adaptations: hypertrophy, hyperplasia, atrophy, metaplasia, and dysplasia. Atrophy. It is the shrinkage in cell size by the loss of cellular substance. With the involvement of a sufficient number of cells, an entire organ can become atrophic. Causes of atrophy includes decreased workload, pressure, blood supply, nutrition, loss of endocrine stimulation, and aging. So, it is decreased size and weight of an organ due to decreased size and number of its component cells. So, here is a shrinkage of kidney which is shown in this picture. Next is hypertrophy. It is an increase in cell size by gain of cellular substance. So, muscle hypertrophy is an example of hypertrophy. With the involvement of sufficient number of cells, an entire organ can become hypertrophic. Hyper term is related to excess of something. So, hypertrophy is caused either by increased functional demand or by specific endocrine stimulation. Not only the size but also the phenotype of individual cells can be altered in hypertrophy. With increasing demand, hypertrophy can reach a limit beyond which degenerative changes and organ failure can occur. So, here is the example of muscular hypertrophy after a regular exercise. So, necrosis: microscopically, cell membrane disappears, cytoplasm is swollen, mitochondria is swollen, rupture, and form myelin figures and may be calcified. While nuclear changes in this pycnosis occur, it means nuclear shrink and increase the basophilia. Next is karyoexis. It means the fragmentation of the pycnotic nucleus. Means nucleus is breakdown into big parts. In karyolysis, these big parts break down into smaller ones. Hyperplasia. Hyperplasia constitutes an increase in the number of indigenous or naturally growing cells in an organ or tissue. The enlargement of an organ or tissue caused by an increase in the reproduction rate of its cells. It is often a predisposing condition to neoplasia. Here is an example of prostatic hyperplasia. Metaplasia. It is a reversible change in which one adult cell type is replaced by the another adult cell type. Because of this, normal protective mechanisms may be lost. Persistence of signals that result in metaplasia often lead to neoplasia. So, metaplasia is the replacement of one tissue by the another. For instance, a change in epithelia from columnar to squamous, both these tissues have normal structure, but metaplasia alters the functional capacity of the tissue. For example, in case of cigarette smoking, the ciliated columnar epithelium of bronchial tree of lungs replaced by the squamous epithelium. Next topic is cellular injury. Ability of body to cope up with stress is called adaptation. When cells are stressed so severely that they are no longer able to adapt is called cellular injury. Or when cells are exposed to damaging agents. Under physiological stress or pathological stimuli, cells can undergo adaptation to achieve a new steady state that would be compatible with their viability in the new environment. If the injury is too severe, affected cells die and it's called irreversible injury or cell death. Causes of cellular injury. There are various causes of cellular injury like hypoxia, ischemia, genetic, nutritional imbalance, endocrine, physical agents, chemical agents, infective and related to immunity. So in hypoxia, there is a deficiency of oxygen. Ischemia deals with the less blood supply or decrease in blood supply to various organs. Genetic deals with the gene defects and chromosomal abnormalities. Nutritional imbalance, in this iron and vitamins are responsible for cellular injury. It means imbalance of these type of nutrition can cause the cellular injury. In endocrine, deficiency or excessive hormone activity can also lead to the cellular injury. In physical agents, mechanical trauma, thermal damage, radiations can cause cellular injury. Chemical agents include metal, solvents, drugs. Infective means infection by viruses, bacteria, parasites and other organisms. In immunity, it is caused by the immune system damage. For example, autoimmunity can also cause the cellular injury. Mechanism of cell injury, general principles. First one is loss of energy. It is by ATP depletion and oxygen depletion. So, by the decrease in oxygen supply, the electron transport chain cannot perform its function properly and this will lead to the cell injury. Second one is mitochondrial damage. So, mitochondrial damage causes the leakage of pro-apoptotic molecules which can cause cell injury. Third one is loss of calcium homeostasis. In this, there is increase in calcium entry caused by the increased membrane permeability or by the activating certain enzymes. Fourth one is defects in plasma membrane permeability. This is by the membrane damage, and by membrane damage, cell injury can occur. Next one is generation of reactive oxygen species. They can damage the lipids, proteins, DNA and cause cellular injury. Misfolded protein can promote the apoptosis and this will cause cell injury. Pathogenesis. Mitochondria is concerned with cell respiration and the production of ATP, which is responsible for important vital functions of the cell like cellular osmolarity, membrane transport process and protein synthesis. So, decreasing ATP will cause defect in these processes of our body. So, pathogenesis of cell injury. Mitochondrial oxidative phosphorylation is disrupted first, so this will decrease the ATP, and by decreasing ATP, this will decrease the sodium-potassium pump. If this pump is not working properly, this leads to the accumulation of sodium ions and water in the cell, which can cause cell swelling. Second is altered metabolism. Altered metabolism causes the depletion of glycogen. Third one is lactic acid accumulation. Lactic acid accumulation causes the decreased in pH and increased intracellular osmotic pressure and accumulation of water, which will cause cell swelling. Now the morphology of cell injury. Morphology of reversible cell injury includes cellular swelling and fatty change. And morphology of irreversible cell injury includes autolysis, electrolysis, necrosis, and apoptosis. Cell swelling. It is a reversible cell injury. In this, there is a cloudy swelling and hydrophic change in the cell. Here we can say normal cell and reversible cell injury. In reversible cell injury, there is swelling of endoplasmic reticulum, mitochondrial swelling, dispersion of ribosomes, generalized swelling, clumping of nuclear chromatin, and so on. So, intracellular accumulation. Here is a fatty change. It is abnormal accumulation of intracellular neutral fat that occurs in parenchymatous organs, most commonly liver and heart. Here we can see here is a normal cell and cell of a fatty liver. After abnormal metabolism, there is accumulation of neutral fat in fatty liver. Causes of fatty liver. Alcohol abuse, diabetes mellitus, obesity, various type of drugs, pregnancy, toxins are causes of fatty liver. Other lipid accumulation, cholesterol and cholesterol esters. In case of arteriosclerosis, cholesterol accumulates in smooth muscle cells, and in hereditary hyperlipidemia, cholesterol accumulates in macrophages, usually under the skin, and forming a tumor-like structure known as xanthomas. Accumulation of proteins. In epithelial cells of proximal convoluted tubules of kidney and in plasma cells, there is accumulation of protein. In kidney, the excessive accumulation occurs due to the leakage of protein from glomeruli into the glomerular filtrate. Glycogen accumulation includes glycogen infiltration and glycogen storage. Glycogen appears as a clear vacuole in the cytoplasm of cells. Hyperglycemia also causes the accumulation of glycogen. Accumulation of glycogen is seen in liver and cardiac muscle fiber. So, next topic is cell death. Cell death includes apoptosis and necrosis. As we discussed in previous slides, apoptosis means suicidal cell and necrosis means murder of a cell. So in this diagram, we can see there is a normal cell and normal cell undergoes necrosis in this way and apoptosis in that way. Difference between necrosis and apoptosis. Death of group of cells within the living tissue is called necrosis. Single cell death, which is programmed, is called apoptosis. Causes of necrosis are always pathological, and in case of apoptosis, they may be pathological or physiological. In necrosis, cytoplasm swells, and in apoptosis, it shrinks. There is inflammation in case of necrosis, and there is no surrounding inflammation in case of apoptosis. ATP depletion, membrane injury and free radical damage is the mechanism of necrosis, while gene activation, endonuclease and protease is the mechanism of apoptosis. Pathogenesis of necrosis. There is irreversible mitochondrial dysfunction which is caused by decrease in ATP. So, this will cause increased level of intracellular calcium and which will activate many enzymes like protease, phospholipase and endonuclease. So, this is the diagram of pathogenesis of necrosis. Here is injurious agent which will cause the increased cytosolic calcium entry from the mitochondria as well from endoplasmic reticulum, and this will cause activation of few enzymes like ATPase, phospholipase, protease and endonuclease. ATPase will decrease the ATP, phospholipase decrease the phospholipids, protease disrupt the membrane and cytoskeletal proteins, and endonuclease damage the nuclear chromatin, and this will lead to the cell injury and cell death. Types of necrosis. There are 5 types of necrosis: coagulative necrosis, liquefactive necrosis, gaseous necrosis, fat necrosis, fibrinoid necrosis. Coagulative necrosis is the most common type of necrosis. In this, there is coagulation of protein of cells. Protein denaturation predominates the enzymatic digestion. Next is liquefactive necrosis. Liquefactive necrosis is due to the enzymatic dissolution of necrotic cells. In this, necrotic tissue is liquefied. Third one is gaseous necrosis. Usually it is seen in infections like TB and fungal infections, and it has cheese-like appearance. Fat necrosis. Fatty acid binds with the calcium from the blood to form a calcium source. The process called saponification, and it gives the chalky-like appearance. Last one is fibrinoid necrosis. Damage to the wall of the blood vessels causes the fibrinoid necrosis. Next one is apoptosis. It is a type of cell death referred to as single or programmed cell death. It is important mechanism for the removal of cells as occur in cells with irreparable DNA damage, like from viruses, free radicals, chemicals, etc., and protecting against neoplastic transformation. Apoptosis is from 2 types, physiological and pathological. In physiological, menstruation, menopause, embryogenesis, immune tolerance comes under physiological processes. And in pathological processes, acute inflammation, organ atrophy, neoplasia, graft rejection and viral hepatitis can cause the apoptosis. Next topic is calcification. Pathological calcification is the deposition of calcium salts in sites other than the bones and teeth. It is of 3 types: dystrophic calcification, metastatic calcification and stone formation. Dystrophic calcification occurs in non-viable tissue with normal blood calcium level, and metastatic calcification occurs in viable tissue and it is caused by hypercalcemia. Dystrophic calcification. Abnormal calcification in which there is a normal blood calcium level plus diseased or necrotic tissue like fat, coagulative, liquefactive and Cajun necrosis. Arteriosclerosis and damaged aging heart valves also causes the dystrophic calcification. In metastatic calcification, there is increased blood calcium level plus normal tissue. In this, there is increased parathyroid hormone secretion, bone destruction, vitamin D disorders, excessive calcium intake as in milk can cause the metastatic calcification. Last topic is electrolyte imbalance. It is the abnormality in the concentration of electrolytes in the body. Electrolytes play an important role in maintaining the homeostasis in the body. Electrolytes like salt, potassium, calcium perform a variety of important functions within our body. It can be caused by losing the fluid as a result of persistent vomiting or diarrhea or sweating. Severe electrolyte imbalance can cause serious problems like coma, seizures and cardiac arrest. The symptoms of electrolyte imbalance can include twitching, weakness, and if unchecked, seizures and heart rhythm disturbances. Thank you, my dear students. Wish you all the best.